The overall goal of this procedure is to present a model of cellular transplantation based on the use of olfactory ensheathing cells in a laryngeal nerve injury model. This is accomplished by first sectioning and performing anastomosis of the recurrent laryngeal nerve. Next, the olfactory ensheathing cells, obtained from olfactory mucosa or olfactory bulbs, are transplanted. Then, 2 to 3 months after surgery, the animals are evaluated by laryngoscopy and electromyography. Finally, the recurrent laryngeal nerve is isolated for histological studies. Ultimately, results can be obtained that show olfactory ensheathing cells improving axonal regrowth of the recurrent laryngeal nerve and functional recovery of the vocal cords through laryngoscopy, electromyography, and histological measurements.
This method can answer key questions in the field of peripheral nerve injury that involve the measure of axonal regrowth and the ability to quantify functionality of the recovery.
Generally, individuals new to this method will struggle because recurrent laryngeal nerves anastomosis requires microsurgical skills.
After generating GFP-labeled primary cultures of olfactory bulbs, or OB, and olfactory mucosa, or OM, as outlined in the written protocol, prepare for surgery by autoclaving